I would also like to welcome you to the first of the webinar series for iNEXT-Discovery. Well, we will talk about biophysical methods in the context of macromolecular interactions. I would like to come up first with a few definitions, like biophysics, which is the study of biological processes using the theories and tools of physics. This is a very big field. And actually, all the structural methods that iNEXT deals with, like X-ray, crystallography and scattering, electron microscopy, NMR, and so on, are basically part of biophysics. It's the use of physical methods to study biological processes, or to be exact, biological molecules, the structure in that case. In the context of this talk, we typically mean methods to quantitate molecular properties. For example, the stability or the strength of interaction and so on. And more specifically, in this seminar, I will deal with the methods to examine macromolecular properties and macromolecular interactions specifically in vitro, because of course lots of these methods can be done in cells, which is a huge and complicated field. Even more specifically, I will focus on the methods that are available on iNEXT-Discovery, and I should mention here that there are more methods that are available to most of you that are listening, and the easiest way to access them is through Instruct-ERIC, that has more sites that offer biophysical methods, but also different methods. Last but not least, I should say that if you wish to do these experiments with us, you need purified and soluble components of all experiments. That is the recombinant protein. Of course, you can buy the DNA, you can buy the ligands, the lipids, the carbohydrates, but you should have all the molecules at the point that you can start the experiment with us. Again, if in particular you do not have purified protein, Instruct-ERIC is a way to go, because you can apply and access one of the production sites or contact directly the protein facility at the NKI or other facilities, and actually we can help you, especially through Instruct, to make purified protein and later access biophysical methods for studying macromolecular interactions. The first method that I wanted to discuss is multi-angle laser scattering, which is a very simple but important method that's mostly the first characterization step, but also as a way to study the interaction between macromolecules. So the idea here is that there is a size exclusion system, gel filtration as we call it, column, that at the end of it, instead of only coupling the UV like you would have in a typical system, we couple the light scattering detector, which is typically multi-angles or at least 2 or 3 angles, and a refractometer, which is another way of measuring the concentration, and all these signals are recorded to a computer, which end up with what you see, for example, in the right, that you see like the UV signal, which is the curves, but you also see the more horizontal lines. That is the calculation based on light scattering of the molecular weight of the complex. As these pass the gel filtration, we can do a lot of things. So you can see, for example, in a magenta light, to the left we have something that looks a bit bigger. To the right we have something that looks a bit smaller. So perhaps there is a gradient of the stoichiometry of this complex along this single peak. But you see this is less pronounced on the blue or the red line, which in that case it's two different antibodies that seem to behave differently. And you see, for example, in that case, there's a fourth sample that comes later in the column. It still has the same molecular weight as the first 3 samples, and perhaps it comes later in the column because of a shape difference or a charge difference that might be interfering with the matrix of the column. The second biophysical method that I want to talk about is the thermal shift assays. And the first and the most popular actually way through the years to do thermal shift assays is the so-called thermoflow that we typically use an external reporter, a dye. In most cases, people use cyproorans, which has some properties that it likes to bind hydrophobic areas in your protein, and then it quenches, and then the fluorescence changes. So as in all the thermal shift assays, you put your protein or your sample, whatever it is, and you increase the temperature. And in that case, the cyproorans dye will bind to the protein as it's unfolding, the fluorescence will increase, you will reach a peak, and later, as the protein aggregates and the hydrophobic surfaces are covered because of aggregation, then the fluorescence will decrease. The advantage of this assay is that it needs relatively little protein, and it's relatively easy to do and only requires an RT-PCR machine. And it is very useful for checking different buffers quickly, or the binding of ligands or mutants or anything like that. Another type of thermal shift assay is the so-called nanodifferential scanning fluorimetry, that these days is also known quite a lot by the brand name of one of the instruments, which is the Prometheus. So the idea here, instead of having an external reporter, you're actually using intrinsic fluorescence of your protein from tryptophan typically, and you follow tryptophan fluorescence as your protein unfolds. You have two advantages. This does not depend on an external reporter. Most proteins have tryptophan, not all of them. The disadvantage is that it needs specialized equipment. It's not as high throughput as the thermoflow, which you can do in 48 well plates. This you can do 16 of these capillaries at the time. But actually, it's pretty quick. And we have exemplified together as others that this technique can be used also for ligand binding screening, because ligands change the TM during binding, but it's also a very cheap and quite fast technique for screening fragment libraries. And Mishba Uddin Ahmad in my group has shown that we can screen this library within 3 days, basically 768 compounds, and get hits that are quite complementary to X-ray crystallography and NMR approaches. A very sophisticated technique that I should say it's something that we're very good at, mainly due to the presence of Alexander Fish, who is actually in charge of all the biophysics methods in our facility, is the surface plasmon resonance, which is another method that is known by its brand name, very often Biacor. And we actually do have a Biacor T200 instrument for quite a number of years now. It works really, really well. And here the idea is that you're measuring the mass transfer with no kind of labels or reporters or anything as well. The catch in this technique that one molecule needs to be immobilized. And this is immobilized in a specific metal surface. This is typically gold. And then you have a light source, which is in a very narrow angle, not like in that scheme. It hits the metal surface in such an angle through a glass prism that it creates a phenomenon called plasmon resonance. And this phenomenon of the plasmon resonance shifts the intensity minimum, so you can use a detector for the reflected light, also at a small incident angle, and measure the signal which is proportional to the mass. So you can imagine that if I go there and I immobilize any biomolecule, which is the one that I need to immobilize, I can do that by amino coupling, I can do it by antibodies, I can even do it by histag. There are many methods to do that. And then you can flow an analyte. And it's important actually that you do that in flow, and flow is one of the parameters you optimize. And then not only by injecting increasing amounts of analyte, you can calculate the affinity, the KD of a complex, but you can also measure kinetics if they're slow, if they're slower than 1 or 2 seconds. And actually quite a lot of reactions that we're interested in, they're slower than a second more or less, and you can measure it. So you can measure the K on, how fast things bind, and the K off, which is a very important parameter, which is independent of the concentration of analytes, and it's actually very characteristic of many interaction. It's directly correlated to the half time that the complex spends together. And I should say that as part of the offer is that we help you to design the best experiment, what to immobilize, for example, how to run it. We also offer the reagents in that case as part of the service. And this is a technique that very often is tricky to set it up quickly, but I think I can convincingly say that that's something that we have really very long expertise on. So another category of methods that are very common in many labs, but we also have them available, are in general fluorescence methods. So in fluorescence methods, for the non-experts, the idea is that you have something that has a fluorophore, so you need a label. At least one of the molecules that you're dealing with must be labeled. And then you throw light on it, you get excitation of electrons. At some point, you get loss of energy, and this comes up with light emission, and you measure the emitted light. So it's absorption, excitation, emission. And that is your fluorescence signal. So the nice thing with fluorescence is that extremely versatile modes of measurement. So you can measure many different things in slightly different ways. So just to be sure that we are all on the same page, excitation happens in a specific wavelength. So the arrow there is the wavelength that typically you get on the catalog, that this fluorophore is excited on that specific wavelength. However, do not forget that there is a band pass. There's some kind of curve, and depending on the fluorophore, it might be excited a bit at a lower wavelength or a bit at a higher wavelength. But of course, there is typically a peak. Fluorescence emission also happens in the same way. There is a maximum of the emission, but this is also a spectrum. It has the tendency to be bleeding a bit towards the higher wavelength, towards the lower energy, but it depends also sometimes on the fluorophore. And what is important also to realize is that regardless of how exactly you excite a certain fluorophore, so you see like the ABCD points, the emission, the shape of the emission spectrum and the maximum of the fluorescence of the emission spectrum will not change. It will remain the same, but the intensity might change. This is very important property for setting up your experiment, but also for being able to design experiments. So the most typical way of doing fluorescence is the so-called quenching. And in that case, you have a reporter and a quencher. So in that case, you can imagine that they are in the middle of, they're on the two ends of something, which let's say it's a piece of DNA, and then you get enzymatic digestion, and then the reporter is not quenched anymore, and you get a signal. There are many setups like that. You have reporters that are substrates. For example, we use their phosphodiester analogs that are very popular, but you can design, or you can ask your chemistry friends to design such reporters. Perhaps the most common type of measurement in fluorescence for macromolecular interactions is the polarization, which basically you're measuring the anisotropy of the fluorescent signal. So although it's the most used one in vitro, it's quite tricky to understand, but I think it's worth it. So typically, you have a source that emits unpolarized light, and you insert after your source a vertical polarizer filter. And this vertical polarizer will only allow to get vertically polarized light. So what happens then is that if you have a small ligand that is labeled alone in solution, or the ligand in a complex with a significantly bigger other partner, which is very common in complexes, these have different properties. The small ligand is rotating very, very, very fast in solution because it's small, while the big complex is rotating very slowly. So the small ligand will depolarize the light, but the complex, because at the time of the measurement, it's way slower, it will still leave a significant portion of the light polarized. So then you insert, before you read out, a vertical and a horizontal polarizer, and you measure how much of the light remained vertically polarized and how much got depolarized, and what is your measurement for the horizontal polarization. And the ratio between these two signals is the measurement of polarization. You can make some corrections, actually mathematical corrections, and you can measure the anisotropy. The difference is that the anisotropy is independent of the relative masses, so it's a better thing to report. Although I'm very much afraid that people are reporting this interchangeably. So that's something very common, because you can have, for example, a piece of DNA and the transcription factor that binds it that will change polarization. You can have a fluorescent ligand that your protein binds, it will surely change polarization. And that's actually a nice trick. If one of your ligands is fluorescent, you don't need to synthesize all your other ligand fluorescence. Let's say you have a peptide and the protein that binds a peptide. You need one fluorescently labeled peptide. The other peptides can be used as competitors, which will get rid of the peptide that binds the one, and you can measure again the gain in fluorescence as you're competing with ligand. Actually, that's always when you have a label, we tend to forget that, but all the 60s and the 70s biochemistry was actually based on competition. So, and it's of course, it's much cheaper to do the competition. Another major way of measuring fluorescence is the Forster resonance energy transfer, or fluorescence resonance energy transfer. And here the principle is also simple. You know that most likely from your cell biology experience, the ones of you that studied biology at least, so you have light. Let's say it's green light, it emits a certain fluorophore, and it emits red light. And then you have a blue fluorophore that when it's hit by green light, it does not emit light, because the excitation spectrum for this fluorophore is not on the green regime. However, this blue fluorophore, when it hits by red light, it can emit blue light, right? So now, if we put these two fluorophores are far from each other and we only use green light, we get the green light coming on the red fluorophore, red light emitted, but because the blue is far away, we don't get red. When the blue comes close to the red, and so the radius for which you get fluorescence is a few tenths of angstroms, that it falls off with a distance. It's also shape and orientation dependent of the fluorophore, which makes it more complicated. But at the end of the day, you get the green light, the red light is emitted, and if your second fluorophore is close, you get blue light. And then if the red light, if the three spectra are well separated, they will never be completely separated, but if the tails and the peaks, you play with what you measure, you find a window of measurement, you can emit your blue light, and you can see when the two things come together. There's a lot of modes for measure that. You can have a sensory domain and a substrate. Then when they meet, you get FRET. You can have a sensory domain that binds a ligand, and then and the C terminus come together, and then you get FRET. Another mode is that you have a multi-domain protein that under certain conditions, one domain meets the other, or it's because of the substrate got there, and then you get FRET. And you can also have the opposite. You have a FRET couple that is giving FRET because it's, for example, the N and the C terminus of a peptide or of a DNA that's cleaved. You cleave it with a protease or a DMase, it doesn't matter, and then you lose FRET signal. So this is limited many times just by your imagination. People are typically using GFP for the N and C termini, but there are methods that you can chemically label the N and the C terminus. The sortases is one of the methods that this can be done chemically. And then you have small fluorophores that you can measure your signals very well and very accurately. A very particular type of FRET that I'm mentioning because we are able to do, and it is very useful, is the so-called time resolved FRET, or TR FRET. TR FRET is using a specific family of caged fluorophores that actually Chancellor Merkel did her PhD in one of those in a European compound, but never mind. And what happens here is in the beginning, like you get your FRET signal or the lack of FRET signal, but very often you have in the same wavelength a lot of background. That's because of your buffer, it's because of the ligand. You don't know if it's because of the ligand. But what you know is that the signal can fall off with a certain time pattern. So if you time your delay and your measurement window, it gives you if you time your energy pulses and the measurement window, you can actually isolate the background from the real signal. There are excellent antibodies for this, and actually it's a very interesting method for screening very big libraries of compounds, because big compound libraries that tend to have a lot of molecules that have autofluorescence, and they give you background signal and you think they're hits on your assay. So this is a very good way, if you set it up, to get rid of this intrinsic fluorescence in big libraries, but it has other advantages, and it's very sensitive and a very good method of measuring. Something I want to mention specifically in fluorescence is that you can have two very different setups. To the left, it's one of our fluorimeters. It's the Ferrastar. That's a high throughput fluorimeter. We can do assays at 1536 well plates, which we can even pipette robotically or with a multi-pipette, it doesn't matter. But typically there, what you do in the fluorimeter, although it has an injection mode, it has an injector, and you can initiate and measure reactions. You can resolve things at the level of several seconds realistically. Even if you only do a single measurement, you inject, you still have a delay in what you're measuring. Perhaps you can measure in the second rate. But in general, what you do is that you have your fluorescent important, and you are titrating increasing concentrations of the second component. And then you plot your signal as a function of concentration in general. And from the shape of this curve, you can fit it and find the affinity, right? So you use one ligand, multiple concentrations of different things, you find the affinities for this ligand, or you do competition or whatever. A setup that we have, and I think it's also pretty unique in many labs, is a stopped flow in which we can mix rapidly the reagents, and then we can look at how the signal develops over time. So because of the mixing and because everything is optimized and so on, the great thing is that we can measure transient kinetics of events that are happening after 5 milliseconds. Typically, actually, you stop almost measuring after a couple of seconds. So these events are, of course, the best way to really understand the system in terms of kinetics, but also you can study this way conformational changes. Getting towards the end, a new method that has also been very popular is the microscale thermophoresis, or the MST, often known, so that's a machine, the monolith that introduced and popularized the method. So here the idea is, again, you need to label, but the idea is basically you have a gain and concentration series, you heat up a capillary, the molecules are trying to escape from the heat, and from the way that they escape, you can measure how the concentration changes, and you can plot the signal. That's typically the thermophoresis signal in that region. And this way, you can also understand the affinity between components. A very old method, historically, that's the first thing that we set up in the lab almost 20 years ago, is the so-called isothermal titration calorimetry. In this case, you are titrating two components, for example, a protein and the ligand. And what you do is that you do that in a chamber in which you can keep the temperature constant. So that's why it's isothermal. So you inject a small amount of ligand and you measure the amount of energy that the system has to put to this chamber to keep the temperature constant. Then you put a bit more ligand and you measure again the energy. So this way, because you are doing things in terms of energy, you do not only get the affinity and the stoichiometry, the KD, and what the stoichiometry of the reaction is, but you can actually estimate pretty well the enthalpy, and because of the constant temperature, you can estimate the entropy, and plus so that separates the free energy of this reaction to the enthalpic and entropic components, which are sometimes key to understand the way that lead molecules function, and in terms of their interaction with your target. For example, there are also some much more imaginative applications. So that's my last slide. I was a bit over time, which is not a good example for the next speakers if they're listening. But what can you do with all these things? You can ask us to find out the affinity of known small molecule ligands like DNAs or lipids protein. You mentioned it. You can measure the stability of a wild type protein and mutants to extract conclusions about the behavior of these mutants. You can find the affinity between two proteins or a protein DNA interaction. We can help you if you have equilibrium measurements by fluorescence to transfer that to transient kinetics. That's something we would love to see projects like that. We can help you set up any of these assays for future screening for ligand libraries and so on. And also specifically, we can help you screen the DSI fragment library, that's the Diamond, SGX, and iNEXT fragment library for heats by MST. Or anything else, please feel free to talk to us and we will try to help you. If you want to apply, you can go to the iNEXT website, which you see there. You click on Apply for Access, big red button to the right, select macromolecular interactions, and you can apply. If you want to make protein, you can also do that through Instruct, or actually, if you're in Oncode Institute, we run some specific services for colleagues that have no in vitro experience for making protein. Of course, feel free to contact us and we will help you set up your own assay and your own measurements. And all of this we can also do remotely. So thank you. Sorry, it was a bit long, and I'm happy to take any questions either about what I talked about or anything else about iNext.
Thank you very much, Tassos. I received 2 questions so far. One is from